Welcome to Biology My Passion. I'm Soumya Harikrishna. Today, let's begin a series of previous year's questions chapter wise. Let's begin with the first chapter. An angiosperm embryo sac is located within the placenta, megasporangium, nucellus, ovary. So we know that inside the ovule there is nucellus. One of the nucellar tissues is later becoming the embryo sac. So the appropriate answer here is nucellus. Long ribbon-like pollen grains are seen in some. We know in aquatic plants, ribbon-like pollen grains facilitate the movement in the water. So option A. The part of the ovule that develops into protective coats of a seed after fertilization in a typical flowering plant is post-fertilization event. The protective coat is nothing but the integuments. You know, the ovule has embryo sac surrounded by little bit of nucellus. Over that there are two integuments, inner and outer integuments. So the integuments later become the seed cover. In which one of the following floral plants are many embryos formed in the seeds without fertilization of the egg cell? It is a case of polyembryony. Example of polyembryony is citrus. In which of the following plants are both male and female flowers born on the same plant and the mode of pollination can be geitonogamy or xenogamy? Here spinach is the answer. Then choose the option that gives the correct number of pollen grains that will be formed after 325 microspore mother cells undergo microsporogenesis. We know that every single microspore mother cell or pollen mother cell undergoes meiosis to give rise to 4 daughter cells. So here the number is 325. So multiplied by 4, we will get 1300 microspores. Select the option that shows the correctly identified U, X, Y and Z in the developing dicot embryo. X is the suspensor, Y is the cotyledon, Z is the radicle and U is the plumule. Now a few assertion and reason questions. In coconut, mitotic divisions of nuclei in the embryo sac are free nuclear. Nuclear divisions are followed immediately by cell wall formation. So here assertion is correct, but reason is false because the nuclear divisions first happen in the form of free nuclear division. Cell wall formation begins later. So option C, assertion true, reason false. Ninth question, assertion endosperm is completely consumed during the development of embryo in X-albuminous seeds. You know that X-albuminous seeds, all the endosperms are completely consumed and there is no endosperm available in the mature state. Whereas reason, castor, pea and beans are examples of X-albuminous seeds. But castor is not an example of X-albuminous, it is an albuminous seed. So option C again, assertion is correct, reason is false. Assertion, in angiosperms, the central cell after triple fusion becomes a primary endosperm cell. It is correct because we know that the polar nuclei will fuse with the male gamete to form the primary endosperm nucleus. Meanwhile, the central cell will turn into the primary endosperm cell. Reason, double fertilization ensures that nutritive tissue is formed before the zygote starts cleaving. See, after double fertilization happens together, later only the embryo sac formation precedes embryo development. So here again, assertion is true, reason is false. Identify A, B, C and D in the table given below. Pericarp, part of the plant it represents is the fruit wall or wall of the ripened ovary because ovary wall later turns into the fruit wall. Second, cotyledon in seed of grass family is called. There is a single cotyledon in monocots, like grass is a monocot. Option B, that is scutellum. Embryonal axis C, embryonal axis, that part of the plant it represents is either it is a radicle or because embryonal axis has a upper part called a epicotyle which will terminate into the plumule and lower part called a hypocotyle which will terminate into the radicle. So any of this can be correct, radicle, plumule, epicotyle or hypocotyle. Then remains of nucellus in a seed that has got a very specific terminology, that is nothing but the perisperm. From which end of the ovule and how does the pollen tube gain its entry into the embryo sac of a hibiscus flower? And the second part, state the fate of the male nuclei present in the pollen tube. So suppose this is an ovule, you have the integuments So here there is the opening called a micropyle. So the pollen tube will be coming here with the two male nucleus. So here is the egg, here are the synergids. So it enters into the synergids. There are filiform apparatus which will guide it into the uh, egg cell. And two male nuclei are there. Okay, so one male nucleus will fuse with the egg. So male nucleus plus the female gamete gives the zygote, which is diploid. So this process is called a fertilization or syngamy, whereas there are polar nuclei in the center. So the second one will go and fuse with the two polar nuclei plus the male gamete. We get a primary endosperm nucleus which is triploid. So since three nuclei are fusing, it is called a triple fusion. So since two fusions happening, one is syngamy, one is triple fusion, this process in angiosperm is termed as double fertilization. Name any two outbreeding devices that flowering plants have developed and explain how they help in encouraging cross-pollination. In the textbook, four methods are given. First, stigma and anther are positioned at different levels. Stigma receptivity and pollen release are not synchronized. Either stigma becomes receptive before the pollen release, or the pollen will be released before the stigma becomes receptive. Then self-incompatibility. Self-incompatibility is a genetic mechanism through which the self-pollen are not allowed to grow. Then production of unisexual flowers, which will always encourage cross-pollination. So any 2 points you can write for 2 marks. If it is 3 marks, you may have to write 3 points. In the transfer section of a mature anther given below, identify A and B and mention their functions. So A is the sporogenous tissue and B is the tapetum. Name the organic material present in the outer layer of pollen grain. Pollen grain is covered by the outermost covering called exine. Exine has a very resistant structure or a chemical called a sporopollenin. Then how are pollen banks useful? Pollen grains use their viability after a particular period of time. It varies from a family or species. It is possible to store pollen grains for a large number of species for years in liquid nitrogen, that is minus 196 degrees Celsius. Such stored pollens can be used as a pollen bank. Pollen banks are very useful in crop breeding programs. Explain the process of the development of male gametophyte in an angiosperm. Why is it called a male gametophyte? So cells of sporogenous tissue inside the anther, the sporogenous tissue is here inside the microsporangium. So these cells of sporogenous tissue, they are otherwise called a pollen mother cell or microspore mother cell. They undergo meiotic division or reduction division to form four microspore, and each one is called a tetrad, which mature and dissociate into four pollen grains or male gametophyte. Why they are called male gametophyte? Because they are the gamete producing structures. Inside that, generative cell will form, which divides to form the male gamete. So since it is producing male gametes, it is called a gametophyte. Double fertilization is an event unique to all flowering plants. Explain the process that I explained in the previous question. Syngamy and fertilization of syngamy and triple fusion together. Give a reason for the following. A seed of an orange has many embryos. It is a case of polyembryony. What happens is the nucellar cells surrounding the embryo start dividing and protrude into the embryo sac and form multiple embryos. That is called a polyembryony. Then cashew is a false fruit, but guava is a true fruit. We know that true fruit is formed from the ovary after fertilization. If any other part contributes to the fruit formation, that is called a false fruit. So in cashew, thalamus also contributes to the formation of fruits along with the ovary. Development of fruits after fertilization from the part other than ovary. Guava fruits develop only from the ovary after fertilization, so it is a true fruit. Now it's a differentiating question. Differentiate between perisperm and a pericarp. So perisperm is a persistent nucellus in some seeds, whereas pericarp is the fruit wall, the wall of ovary which develops into the wall of the fruit. So next, syncarpous pistil and apocarpous pistil. Pistil is the female reproductive part. So fused, if they are all the carpels are fused, then it is called a syn. Syn means fusion. Carpels are fused, whereas if all the carpels or pistils are free, then it is called a apocarpus. Then the third is plumule and a radicle. You know, an embryo has got upper part called above the cotyledon attachment point. The part is called epicotyle, which will develop into the plumule, which is the future stem or terminal part of the epicotyle or shoot tip of the embryonal axis. Whereas the lower part is called a hypocotyle, which will terminate into radicle. So that's the future root or terminal part of the hypocotyle or root tip of the embryonal axis. Explain monosporic development of embryo sac in the ovule of an angiosperm. You know that in monosporic development of embryo sac involves the formation of single functional megaspore. That is, suppose this is the megaspore mother cell. This MMC will first undergo meiosis. So that is reduction division 1, and it will form a dyad, microspore dyad. Then after reduction division 2 or meiosis 2, 4 cells will be forming. Out of those, 3 will degenerate, only 1 will become the functional megaspore. So since this megaspore undergoes 3 mitotic divisions to form single 8-nucleate 7-cell embryo sac, which includes 1 egg cell, 2 cynogytes, 3 antipodal cells, and central cell with 2 haploid polar nuclei. So since there is only one functional megaspore, that is called a monosporic development. And this is diagram of a mature embryo sac. This is a simplified diagram which shows the antipodals to the chelacal end, cynogytes to the micropylar end with the egg together called egg apparatus, and there are polar nuclei in the central cell. One of the major approaches of crop improvement program is artificial hybridization. Explain the steps involved in making sure that only the desired pollen grain pollinates the stigma of a bisexual flower by a plant breeder. So there are different steps in artificial hybridization. First is emasculation, because if it is a bisexual flower, anthers have to be removed from the flower bud before the dehiscence, because it will prevent self-pollination. Then bagging, covering the emasculated flower with a bag to prevent contamination from unwanted pollen grain till we do artificial hybridization. Then pollination. When the stigma becomes receptive, the pollen grains are transferred onto the stigma of the emasculated flower from the suitable male flower. Then rebagging, covering of the flower again to ensure that only the desired pollen fertilizes the ovum. So these 4 are the steps of artificial hybridization. Study the diagram given below showing the modes of pollination. Answer the questions. So given diagram shows 3 methods of pollen transfer. What are the technical terms used for pollen transfer methods 1, 2 and 3? So 1 means in the first one we can see that within the flower this is happening. So that is called autogamy. Second is from one flower to the other of the same plant, that is called a geitonogamy. Third is between 2 flowers of 2 different plants, that is called a xenogamy. How do the following plants achieve pollination successfully? One is water lily. In water lily, they are pollinated by insects or wind. Even though they are aquatic plants, the flower is on the surface of water and it is a colorful flower, so it can be pollinated by insects or wind. Whereas in the second case, Vallisneria, female flowers on long stalks reach water surface. Male flowers or pollen are released on the water and carried by water current to female flowers to achieve pollination. Flowering plants have developed many devices to avoid inbreeding depression. Explain one hereditary and one physiological device which helps plants to achieve this target. We explained 4 methods of outbreeding devices previously. So out of that, genetic is the self-incompatibility, that is preventing self-pollen, same flower or other flowers of the same plant, from fertilizing the ovules by inhibiting pollen germination, pollen tube growth in pistil. Whereas physiological, if asked, we can write pollen release and stigma receptivity are not synchronized, that is not simultaneous. Either pollen matures earlier and stigma later, or pollen matures later than stigma. Explain the development of male gametophyte in an angiosperm. Each microspore mother cell in the sporogenous tissue undergoes meiosis, forming microspores tetrad by the process called microsporogenesis. Microspores form clusters of 4 cells, tetrad. As the anther matures, they dehydrate. Each one undergo asymmetric division to produce one vegetative cell and one generative cell. This is the structure of a male gametophyte or the pollen grain. It has an outer covering called exine, inner covering called intine. Exine has sporopollenin. There are areas where sporopollenin absent called germ pore. Inside the cell will divide into two unequal divisions. One is the large vegetative cell, second is the small generative cell. Generative cell nucleus further divides to form the two male gametes. Explain the process of double fertilization in an angiosperm, starting from the germination of pollen grains on the stigma, mentioning the ploidy of the end products formed at the end. State the role of cyanogytes during the course of the process. Why does the development of endosperm precede that of embryo? I already explained the process of double fertilization, but pollen germination is the pollen grain germinates on the stigma, forming a pollen tube that grows through the style and enters the ovule via the micropyle. Synergids, one synergid guides the pollen tube into the embryo sac. Then syngamy and triple fusion together called double fertilization. Zygote is diploid and primary endosperm nucleus or the primary endosperm nucleus or primary endosperm cell is triploid. The endosperm develops before the embryo because it provides essential nutrients for the developing embryo, supporting its early growth and establishment. A label diagram of a fertilized embryo sac of an angiosperm that we already saw a simplified diagram. This is a diagram from the textbook. This is post fertilization because you can see that the antipodals are degenerating and zygote is formed. Explain double fertilization that we already discussed. So this is a repeatedly asked question because these questions I have taken from the previous year question papers. What is megasporogenesis? Trace the development of large megaspore mother cell to mature embryo sac in an angiosperm. So first what happens is the definition process of formation of megaspore from megaspore mother cell. First megaspore mother cell will undergo meiosis 1. I told it will develop into a dyad, then into a tetrad. Then 3 megaspore cells will degenerate and one becomes functional. So it is called a monosporic development. So that functional megaspore mother cell will now undergo nuclear division, that also mitotic division because they are already haploid. This leads to the formation of an embryo sac later with a wall formation and ends up in 8 nuclei and a 7-celled stage. 3 nuclei are grouped towards the chelacal end. 3 nuclei will be towards the micropylar end. The chelacal end, the cells are called antipodals and the 3 nuclei towards the micropylar end are forming the egg apparatus and 2 will remain in the center called the polar nuclei. Then 6 of the 8 nuclei are surrounded by cell wall and organized into cells containing 1 egg cell between 2 synergids and 3 antipodals. The remaining 2 nuclei, polar nuclei, are situated in the large central cell. Hope these questions helped you well. Please like, share and subscribe to my channel, Biology My Passion. Thank you for watching.